Okay, so welcome back to this next video in which we are discussing heterotrimeric G proteins and adenylyl cyclases. Okay, so we're currently in the process of discussing heterotrimeric G proteins. Okay, so we've discussed the huge number of genes that you have for alpha subunits. What we now want to discuss is the genes for beta subunits and the gene that has an extra splice variant that gives rise to a total number of 6 different subunits. Okay, right. So the 5 genes for beta subunits are named very sensibly. Okay, so they are called G beta 1, G beta 2. Okay, so G protein, B, beta subunit, and then the 2. G beta 3, G beta 4, and G beta 5. Okay, so that's the naming of the 5 different genes. Now, most of them just produce one known protein. So, for instance, G beta 1 will produce, the G beta 1 gene will produce the G beta 1 protein. The G beta 2 gene will produce the G beta 2 protein. The G beta 3 gene will produce the G beta 3 protein. The G beta 4 gene will produce the G beta 4 protein. And then finally, G beta 5 then produces 2 different splice variants. Okay, so G beta 5 also produces G beta 5 and G beta 5L. Okay, so this is the naming of the 2 different splice variants. There is G beta 5 is one of the splice variants, and G beta 5L is the other splice variant. So these then now are the 6 different subunits that can fill the place of the beta subunit in heterotrimeric G proteins. Okay, now the final thing we need to discuss is the gamma subunits of heterotrimeric G proteins. Okay, so this one's simple because there are no splice variants, at least not that are known. Okay, so there are 12 different genes for gamma subunits, and there are 12 different subunits. Okay, and again, the naming is very nice and simple. Okay, so you have the gamma 1 subunit, okay, and I should put G gamma 1, then you have G gamma 2, and then it goes on all the way up to G gamma 5, okay, so G gamma 1, G gamma 2, G gamma 3, G gamma 4, G gamma 5, and then there's a blip. You miss out G gamma 6, there is no G gamma 6, and then there is a G gamma 7, and then it goes on all the way up to 13, G gamma 8, all the way up to G gamma 13. Okay, so if there hadn't been the blip at 6, then obviously it would have gone from G gamma 1 to G gamma 12. You miss out 6, and therefore you have to go up all the way to 13. Okay, so that overall then gives us 12 different gamma subunits. So overall there are a huge number of different heterotrimeric G proteins that you can build. Okay, now strictly speaking, when people name heterotrimeric G proteins, what they should do is they should tell you exactly which alpha subunit they're talking about, exactly which beta subunit they're talking about, and exactly which gamma subunit they're talking about. However, people rarely do this. So for instance, an example would be G alpha I1, G alpha beta 1, gamma 1. This would be an example of the naming system for a heterotrimeric G protein where you name exactly which alpha subunit, exactly which beta subunit, and exactly which gamma subunit are within that heterotrimeric G protein. Okay, but people rarely do name heterotrimeric G proteins like this. Instead, they often just tell you which of the 4 families the alpha subunit is in. So usually they just name heterotrimeric G proteins according to which alpha subunit you have. And sometimes people don't actually even tell you the specific alpha subunit, because often people don't even know which specific alpha subunit they're looking at. Okay, so often people will just tell you which family the alpha subunit is in. So, for instance, this might just be called GI/0. Maybe if they knew that it was specifically an I alpha subunit, i.e. it's alpha I1, then they might just call it GI, but often people will just call it GI/naught, because alpha I subunits and alpha naught subunits have very similar functions, and certainly the three alpha I subunits, G alpha I1, G alpha I2, and G alpha I3, those certainly have very similar functions, okay, so it's quite difficult often to tell which one is specifically doing what you think, and often it might be that all of them are doing all of them are being activated by the G protein coupled receptor, and they're all doing this activity. Okay, right, so it's not uncommon to see people labeling the heterotrimeric G protein according to which alpha subunit they have, and even more generally, just sort of generalizing it potentially to which family it's in. Okay, so let's now look at the activation of heterotrimeric G proteins by G protein coupled receptors, and then we'll look at the downstream activity of some of these heterotrimeric G protein subunits. Okay, right, so let's draw our G protein coupled receptor. We'll draw it here. Okay, so here is our 7 transmembrane receptor with these 7 membrane spanning alpha helices, like so. So here's the amino terminus, here's the carboxylic acid terminus, and what's going to happen is the ligand is going to come in, it will bind to residues on the membrane spanning alpha helices. Now what does that trigger? Well, it triggers a conformational change in the G protein coupled receptor. And where is this conformational change occurring? Well, it occurs on these 3 intracellular loops. So you'll notice that in this structure you have these 3 intracellular loops. You have one between the membrane spanning alpha helix 1 and the membrane spanning alpha helix 2, one between the 3rd and the 4th membrane spanning alpha helix, and one between the 5th and the 6th membrane spanning alpha helix. So I've highlighted them all in turquoise. And these are called the intracellular loops. An intracellular loop is abbreviated to ICL. So this one will be called ICL1 for intracellular loop 1. This one would be called intracellular loop 2 for it would be abbreviated ICL2, and this one ICL3 for intracellular loop 3. Okay, and basically what will happen is the ligand will bind, that will induce a conformational change in the intracellular loops, and now what will happen is that conformational change will then allow heterotrimeric G proteins to bind the G protein coupled receptor at these intracellular loop regions, which are obviously the main portion of the intracellular domain of the G protein coupled receptor. So basically these intracellular loops, once they've undergone the conformational change, will produce a binding site for the heterotrimeric G protein. Okay, so which portion of the heterotrimeric G protein binds there? What's specifically the alpha portion? Okay, so the alpha subunit binds, however, it cannot bind unless it's also got the beta gamma subunit bound to it, and unfortunately the coup that I've written there is in the way, so I can't actually show them next to each other, but they should be next to each other. Okay, so the alpha subunit binds to this binding pocket that is created by these intracellular loops of the heterotrimeric G proteins, but it can only do this if it has the beta and the gamma subunit with it, and it will only be bound to the beta and the gamma subunit if it is in the off state and therefore has guanosine diphosphate bound to it. Okay, so off-state heterotrimeric G proteins can bind to this intracellular loops of these activated G protein coupled receptors, and specifically it's the alpha subunit that binds. Now, specific G protein coupled receptors will bind different alpha subunits, okay? So, for instance, the H1 receptor, which is an example of a rhodopsin family G protein coupled receptor, okay? Its ligand is histamine, and when histamine binds, it will undergo a conformational change, and it will then interact with heterotrimeric G proteins. Now, the alpha subunits that it likes to bind are specifically the G-alpha Q subunit, so it won't bind to heterotrimeric G proteins that don't have a G-alpha Q subunit. Okay, so G protein coupled receptors will be specific in which alpha subunit they want to bind. And remember, for the alpha subunit to bind, it's essential that you have it bound to the beta and the gamma subunit. And for that to occur, it needs to be in the off state with GDP bound. Now what will happen is the binding of the alpha subunit to this binding pocket on the intracellular domain of the G protein coupled receptor will trigger the release of guanosine diphosphate from the alpha subunit of the heterotrimeric G protein, and instead guanosine triphosphate, GTP, will come and bind in there. Okay, now when GTP binds the alpha subunit, it breaks away from the beta and the gamma subunit, so they cleave apart. Okay, and now once the alpha subunit no longer is bound to the beta gamma subunit, basically the beta gamma subunit will wander off. Okay, and also the intracellular loops of the G protein coupled receptor, they will lose their affinity for the alpha subunit because they only wanted to bind to the alpha subunit when it had GDP bound. Okay, and also when it had the beta gamma subunit attached to it. So once they break apart because of the GTP binding, the alpha subunit will also break away from the G protein coupled receptor. So the overall effect of this is that you end up with two subunits, basically, that are now capable of having regulatory effects on the adenylyl cyclases, as well as many other proteins as well. Okay, so you end up with an alpha subunit which has GTP bound to it, which is still attached to the inner leaflet of the phospholipid bilayer by this myristoyl or palmitoyl group or both. Okay, and then you also end up with the beta gamma complex here, which has a beta subunit bound to a gamma subunit here with a prenyl group of the gamma subunit. Okay, so this is the beta gamma, and I think it should be called a complex rather than a subunit. Okay, so you produce the alpha GTP subunit and also a beta gamma complex. Okay, right. And these two protein subunits are then going to have downstream effects. Okay, and we're specifically interested in their regulatory ability on adenylyl cyclases. Okay, so let's begin. Okay, so the first thing to say is that if your heterotrimeric G protein that has just been activated is a heterotrimeric G protein which has either the G-alpha S subunit as its alpha subunit or the G-alpha OLF subunit as its alpha subunit, then these subunits both activate absolutely all adenylyl cyclases. Okay, so G-alpha OLF is very, very similar to G-alpha S. Okay, it's found in, for instance, the olfactory epithelium in a greater expression level than G-alpha S is found in the olfactory epithelium. Okay, so basically it just replaces G-alpha S in the olfactory epithelium and in a few other locations as well. So this is either alpha S or it's alpha OLF. Okay, and basically it's now got GTP bound to it, and it's going to activate all 9 of the adenylyl cyclase enzymes. Now, how does it do this? Well, basically, if you remember, how did we activate adenylyl cyclase enzymes? Let me just draw an adenylyl cyclase here. So remember, adenylyl cyclase enzymes have these 2 transmembrane domains where you have these 6 membrane spanning alpha helices, okay, like so. Then they have the amino terminus here, and then the carboxylic acid terminus down here, and then they have the C1 domain, which is divided up into C1a and C1b, and then they have the C2 domain, which is divided up into C2a and C2b. Okay, and remember, to activate the enzyme, what you need to do is dimerize C1a and C2a. So basically, the way that these alpha S/alpha OLF subunits, which have GTP bound to them, activate the adenylyl cyclase enzymes is they bind to both the C1 domain and the C2 domain. Okay, so they bind to this domain here and to this domain here, and they hold the two domains close together, and then what can happen is the C1a and the C2a combine together and produce an active enzyme which can then convert ATP into cyclic AMP and pyrophosphate. Okay, so by binding to both the C1 domain and the C2 domain, the alpha S/alpha OLF subunit, which has GTP bound to it, promotes the dimerization of the C1a and C2a domains, and hence promotes the activation of the adenylyl cyclase enzymes. And they will do this for all 9 forms of adenylyl cyclase. Okay, right. So we'll call it there for this video, and we'll discuss other alpha and also beta gamma subunits which have a role in regulating adenylyl cyclase function.